Hi, this is Nasim Sanati. I'm a research software developer and bioinformatician from Oregon Health and Science University, and I'm a part of the Reactome's team. So here today I'll be presenting our effort on how we are expanding Reactome functional interaction network to allow for exposure of knowledge space of these understudied proteins in the context of biological pathways. To give a bit of a background and clarify some terminology here, a recent survey of the human proteome done by the IDG has identified a class of proteins known as DARK, which virtually nothing is known about the development or druggability of these human targets. Now, projecting these understudied proteins onto reactome pathways provides a pathway view or process context of the biochemical mechanism of their molecular function. It also provides us with a familiar software environment for the research community to explore, design experiments, and really test our hypotheses to essentially investigate the clinical actionability of these targets. We also know that navigating through the complex biological processes can be too cluttered. And to help the navigation and investigation, we convert these pathways into a pairwise functional interaction network, just like the Reactome F5S application. With all said, to expand our functional interaction knowledge space, we focused on integrating data types from a variety of reliable resources. Starting from the bottom corner of the data triangle here, we pulled protein-protein interactions in non-human species from StringDB, BioGrid, and BioPlex, and mapped them back to human proteome by using the orthologous mappings downloaded from Panther and Ensemble. From Harmonism Gene Similarity Portal, we first manually curated datasets with pairwise relationships, excluded those that were from Reactome, and chose datasets having odds ratio greater than 5 against our own Reactome FIs. And for Coexpressions, we pulled tissue and cancer project RNA-level gene expression from GTEx and TCGA data portals. These samples went through a diligent pipeline converting counts to CPMs, deduplicating, and removing sample outliers, and etc. And we were really pleased to see these beautiful PCAs showing tissue-specific clustering. Then, from these expressions, we constructed Spearman correlation adjacencies of all normal and cancer-specific tissues. And then again, we ensured that the odds ratio for each selected correlation feature was greater than 5 against our own Reactome's FIs. We also pulled data from geobiological sharing that included at least one biological process term. And finally, we looked at domain-domain PFAM to check if two proteins interact via their domain annotation and domain-domain interactions. So overall, we ended with 106 high-quality features from all of these data sources. Now moving on to integrate these evidences and predict functional interactions between proteins, we constructed training and validation datasets with positive protein pairs, or FIs, extracted from annotated Reactome pathways, and negative protein pairs that were just random protein pairs from the whole human proteome. This, of course, creates a classical imbalanced machine learning problem, which was beautifully handled by the training and cross-validating a balanced random forest model that performed pretty well with the area under the curve of 89%. And after getting the prediction probabilities for these FIs or protein pairs, we convert Reactome pathways into Boolean networks and simulate a fuzzy logic to get the impact of these protein classes, especially the dark proteins, onto the Reactome pathways. With all said, you can most definitely utilize this data by visiting our Reactome IDG portal that is actively being developed by Tim Bronson. And he is also presenting his poster at ISMB 2020. Happy exploring, and thank you so much for your time and attention. Please stay safe.